So, as I mentioned last time, this is actually one of my favorite chapters in the book. Unlike the other categories of biomolecules that we talk about, we'll spend multiple chapters talking about the macromolecular form of these building block amino acids. When we link these building block amino acids together, we get something we call polypeptides, or more commonly referred to as proteins. So this chapter is concerned really only with something called protein primary structure. Protein's primary structure is simply the sequence of the amino acids that make up that particular protein. So this would be an example right here of protein primary structure. All right? Remember, when you see something like this, when we have a polypeptide chain or a protein chain, one end of that protein is going to have a free alpha amino group, and we call that the N-terminus or amino terminus of the protein. And then at the other end, we have a carboxyl terminus or a C-terminus, a free alpha carboxyl group. So this has a direction. If I wrote the reverse of this, alanine, glycine, leucine, that would be a different protein than the one I have shown here. Okay? So the fact that we have something called protein primary structure implies that there might be something called a secondary structure. And in fact, there is. There's even a tertiary and a quaternary structure that all refer to more of the three-dimensional shape that particular protein sequence folds into. And we'll talk about those next chapter. Secondary structure has to do with short little segments of the chain, what they fold into. Tertiary structure is how the shape of the overall chain, what that folds into. And some proteins are composed of multiple chains that associate with each other. And how they associate with each other we call quaternary structure. Okay, so just keep that in the back burner. We'll deal with that next chapter. Know that there are these other types. Right now, we're just concerned about proteins' primary structure. Now, why do we make such a big deal out of this primary structure? I've said a couple of times in this class that the shape of a protein, its three-dimensional structure, is really what determines its function. That shape of the protein is going to give it its special properties, its ability to catalyze a reaction or carry out the function that the cell needs it to carry out. Well, it turns out that the protein's three-dimensional shape depends upon the sequence of the amino acids that make up that particular protein. Three-dimensional shape is dependent then upon the primary structure. In fact, what we find is that the characteristics of a particular protein are more dependent on the sequence of the amino acids than the actual amino acid composition. Another way of saying that would be if I took a protein that had a certain number of alanines, certain number of glycines, so on and so forth, and they were arranged in a particular sequence. If I take that same pool of amino acids and made a protein of identical composition, but in a different sequence, in all likelihood, those proteins would be completely different in properties, completely different structure that they would fold into, and therefore completely different function. So it's the sequence that is the most important thing in determining this overall three-dimensional structure and therefore the properties of the particular amino acid. Let's talk just a little bit about the diversity of different types of proteins that we might be able to get using only these 20 amino acids. I mean, you might think that wow, cells have to carry out a whole lot of different functions. And it seems pretty limiting to say that, well, all of these proteins that carry out these vast different functions in the cell, we're only allowed to use these 20 amino acids. That seems very limiting. But think about it this way. If you have even a short protein sequence, let's take something a little ridiculous. Let's say you only have a 4 amino acid peptide, 4 amino acids in length. There are 20 to the 4th power. There are 20 different amino acids. So that means there are 20 to the 4th power different 4 amino acid peptides that you could possibly make. That's about 160,000 different possibilities just for that short little length. Now, if you start taking a typical protein is going to be 100 amino acids long or more. 20 to the 100th power is an astronomical number. Huge, huge, huge diversity possible with just these 20 different amino acids. We can get lots and lots and lots of different types of proteins. Now, remember we talked about that perhaps the primordial molecule of life, or the molecule that really carried out catalytic functions, might have been RNA, right? We talked about that RNA world hypothesis. And you might wonder, well, if that's true, then how come we don't use RNA for that function anymore? Well, one possibility might be this. There are 4 different nucleotides in RNA compared to 20 different amino acids in a protein. So if I have a 4 nucleotide sequence in RNA, that's going to give me 4 to the 4th power, still a pretty large number. I think that's 256, but not anywhere close to the diversity that I can get with the amino acids. Okay? So proteins are going to be more versatile catalysts and likely more powerful catalysts than RNA can be. All right? Now, another point is that, speaking of comparing amino acids to nucleotides as far as what would be an ideal catalytic molecule, with these amino acid side chains, you have lots of different characteristics of those side chains available to choose from. You have some that are hydrophobic, some that are polar and non-charged, some that are positively charged, some that are negatively charged, and so on and so forth. In the nucleotides, each nucleotide looks more or less the same, right? They have an aromatic nitrogen-containing base, ribose sugar, and phosphate. So chemical diversity available is much, much greater also with the amino acids than would be the nucleotides. So all of this is just to show you that, well, even though we don't have RNA playing much of a catalytic role nowadays, likely that's because the amino acids are just so much better at building catalysts than the nucleotides are. Right? Questions on that? Okay. Let's take a look at some typical protein sequences and see if we can come up with some general properties of these that we might observe inside a cell. Most proteins are going to be, there's a wide variety here, but between about 100 and maybe up to 1,000 amino acid units, or what we call residues. The amino acids that make up a protein we call residues. So 100 to 1,000 amino acid residues in length. Now, we do encounter some proteins that are smaller than that, but in general, what we see is that any polypeptide or protein that's shorter than about 40 amino acid residues tends to not fold into a stable three-dimensional shape. In solution, inside the cell, it's going to be flopping around and assuming all kinds of different shapes. That makes it difficult to serve a role as a particular function for the cell. Okay? So usually we see a lower limit of somewhere around 100 or so because of this fact right here, because in order to fold in a particular shape, you need enough amino acids to support that. Now, what about this here? 1,000 amino acids long. That's even a pretty big protein right there. And we don't see many even that large, and much less larger than that, single chains at least. However, we will encounter proteins that collectively are going to be much, much greater than the sum of that 1,000 amino acids in length. What often happens if the cell needs a very, very large protein to carry out very complicated functions or something like that, these large proteins are often composed of multiple amino acid chains. Okay? So you may have 10, 200 amino acid linked chains binding together, associating with each other to make one huge protein. And that is typically the way that cells make very, very large proteins, by making smaller chains that associate with each other. There's actually a pretty practical reason for this. We don't have time to talk about it in this class, but I believe if you take molecular biology, you'll learn how proteins are actually made, how they're synthesized inside the cell. And you'll see that making those proteins requires a lot of energy, a whole lot of energy to make a particular protein chain. And you'll also learn that it's not 100% error-proof, all right? Mistakes can be made. Now, if I have to make a huge polypeptide, let's say 5,000 amino acids in length, first of all, the probability that I'm going to make a mistake is greater, right? There's just 5,000 amino acids, so the probability of making a mistake in one of those is greater than the probability of making a mistake in a much shorter protein. Second of all, if I do make a mistake there, now I have a huge protein that I've spent a lot of energy to make that's likely not going to be useful. So that's a waste of energy for the cell. A more efficient way to do it is to make smaller proteins, smaller chains, and have them associate together to carry out a function that would be equivalent to a much larger protein. Right? Does that make sense? Okay, let's take a look at some of these. And I've jumped ahead to show you some structures of these, some three-dimensional structures. But here's a barely legible list, I think, of some of the proteins that we might encounter throughout this semester. From very small, this is the protein insulin right up here. It actually consists of two very, very small chains that are linked together through disulfide linkages. Insulin isn't a catalytic protein. It actually serves as a signaling molecule, as a hormone. Another small protein that's very important, we'll talk about at the very end of the semester, is this cytochrome C. It plays an important role in electron transport. We'll talk about these two in a little bit of detail. Ribonuclease is an enzyme, a very small enzyme, that breaks up, hydrolyzes phosphodiester bonds in RNA. We'll spend a good deal of time talking about this protein. This is the enzyme lysozyme. You can see that all these are fairly small proteins, right around 100 amino acids or a little over. We'll spend a lot of time talking about this protein. This is a oxygen-binding protein, myoglobin, and its counterpart, much bigger, is hemoglobin here. We'll spend time talking about both of those. Hemoglobin consists of 4 separate chains. Myoglobin is just one chain. Hemoglobin has 4 separate chains. All of them look kind of like myoglobin. Right? This large molecule here we'll actually talk a little bit about in this chapter. This is a protein that's used by your immune system. It's called an immunoglobulin. These ends right here are used to bind to various foreign things that enter your body. So we actually use this in biochemistry research a lot as well. And here's an example of just a huge, giant, large protein. This is glutamine synthase. It's composed of actually 6 separate large chains to make up that one big protein there. Okay, now, we talked a little bit about what can happen with cysteines, right? Cysteine residues in a particular protein can actually form a particular linkage that we call a disulfide linkage. This is a oxidation reaction. Here, the cysteines are in their reduced form. In the presence of oxygen, they can become oxidized and form a linkage to each other. The location of those linkages is actually pretty important for the three-dimensional structure of the protein as well. So, as a result, we kind of consider the locations of any disulfide bonds that are in the protein as part of the primary structure, as kind of associated with the sequence here. Okay? Here's the enzyme ribonuclease that we just talked about on the last slide, and you can see that there are 1, 2, 3, 4 particular disulfide bonds in that particular protein. Notice that they specify this cysteine, cysteine 26, is connected to cysteine 84 there. And if I were to connect this cysteine to a different cysteine in the protein, the enzyme would not be active. It wouldn't carry out its active role. That's because it's not able to fold into its particular shape. We'll see a famous experiment, likely next chapter, dealing with this exact enzyme and showing that is the case. Right? Here's another protein that we talked about earlier. This is the protein insulin. You can see that insulin is composed of two separate chains, and those chains are actually held together by disulfide linkages. So these disulfide linkages are playing a critical role in insulin, because otherwise these chains are probably short enough that they wouldn't stay together very well at all. Insulin also has a disulfide linkage within that smaller chain as well. So disulfide linkages are also considered part of the protein primary structure. All right, let's talk a little bit about the amino acids that we find in proteins and their relative occurrence. So what this table is showing here is this is all 20 different amino acids, and this is a study that was done with 207 different unrelated proteins, and they quantified the percent of occurrence of each of these different amino acids. Now, there are 20 amino acids, so if they're evenly distributed throughout proteins, we'd expect that each to have a percent occurrence of around 5%. Right? And at first glance, you could say, well, they all look about 5%, more or less. They're closer. But if we look at it a little bit closer, we see that there are some proteins that seem to be a little bit more abundant than the others. Take a look at alanine up here, percent occurrence around 9%, almost double what we would predict at 5%. Same thing down here, leucine, glycine, lysine, serine, valine, glutamic acid. All of these we could say are the most commonly occurring amino acids in proteins. Now why might that be? Why would these particular amino acids tend to occur more regularly than some of the other amino acids? Well, I think there's a couple of reasons. First of all, if you look at these, all of these amino acids have side chains that are relatively simple, relatively small, right? That means that it's easier for the cell to make these things. It also means that if you have them in a protein, that amino acid residue is going to be more accommodating. It can fit in a lot of different places in that folded protein, whereas a big giant side chain is going to be more stringent. It's going to be more difficult, perhaps, to fold that into a particular shape, or it's more constraining, I should say. Second thing is, look at this list right here. You have all the different types of amino acids composed in here as well. You have leucine and alanine, which are and valine, which are simple hydrophobic amino acids, right? You have lysine. Lysine's a amino acid side chain that is positively charged at physiological pH. Glutamic acid is going to be negatively charged at physiological pH. And you also have serine, which is a non-charged but polar amino acid. So with that small group there, you've got just about all the chemical functionality that is represented by the amino acid side chains. Right? Now, similarly, if you look at this list, you'll find that some amino acids are very rare, relatively rare compared to the others. Tryptophan down here, only about a 1.1% occurrence. Cysteine, methionine, histine, also amino acids that are relatively rare in proteins. Now, we can probably come up with reasons why these particular amino acids don't occur as readily as well, right? Tryptophan is the largest amino acid. It has this big fused aromatic ring right on it. It's going to be energetically expensive to make, and it's big, right? Takes up a lot of space and everything. We can't imagine having a protein full of tryptophans. One, it would be expensive, energetically expensive to make. The other is those tryptophans, that would seriously limit the conformations that particular protein might be able to fold into because of those big bulky groups there. Why wouldn't you want a protein with a huge number of cysteines in it? Anybody think of why that might not be such a good idea? Exactly, right. These disulfide linkages can be problematic, right? They can really serve a great purpose if the proper ones have formed, but the more cysteines that you have in a protein, the greater the likelihood of the improper disulfide bonds are going to be formed, and that's going to prevent that protein from folding in its correct shape and carrying out its function. Methionine, methionine doesn't form disulfide bonds, but it has that sulfur in it. Sulfur is a big atom, so this is going to be a big, bulky group, a hydrophobic compound that unless we specifically need it, we're probably going to be better off using something like a valine or leucine instead. That would just be a simpler solution. And then this last one here, histidine. This is a special amino acid. One of the unique things about histidine is that it has its side chain, which has a pKa that's pretty close to the physiological pH, the pH that we would typically find inside the cell. That means that histidine can fairly easily oscillate between protonated and positively charged and deprotonated and neutral. So if we had a protein with a whole lot of histidines in it, it would be susceptible to becoming massively positively charged or eliminating that massive positive charge just with small changes in pH. And those charge changes are likely going to drastically change the structure of a protein. So for that reason, we typically don't see proteins composed of huge numbers of histidines. Now, having said that, that same property of histidine makes it extremely useful in the active sites of enzymes where it can act as an acid catalyst or base catalyst, donating a proton to stabilize the transition state of a reaction or pulling off a proton from the substrate to speed up the reaction. So we do see histidine play important roles. We just rarely see it composing a lot of a particular protein. Right. Questions on that? Bless you. Okay, let me give you an introduction now. What we would like to talk about next is how we determine the protein primary structure. How do we know what a particular protein's sequence is? And we'll talk about that later on in the chapter. But before that, this chapter also uses some time to discuss protein purification. In order for us to determine the sequence of a protein, it is often necessary for us to purify it. Inside the cell, that particular protein is present with thousands of other different proteins. We need to isolate that protein we're interested in sequencing away from all of the other proteins that might be inside the cell. So, and in addition to that, one of the key biochemistry techniques in the lab is really purification of these proteins. If we want to learn anything about these proteins, even if we do know the sequence, it's often necessary for us to purify these, to get them isolated from all the other proteins that are present in the cell. Right? Now, this is not a very easy thing to do, right? First of all, as I mentioned, usually inside a particular cell, there's thousands and thousands of different proteins. All of these proteins are composed of amino acids, so they're similar in many ways as well. So picking out the one that you're interested in and getting rid of all the others, that's not a very easy thing to do. Making it more difficult is usually the particular protein that you're interested in is only present in very low concentrations in a particular cell. There may be, maybe 100 copies of that particular protein in a given cell. And that makes it difficult as well. That means that if we want to get any reasonable amount of this protein to study, we have to start with a massive amount of cells, of tissue, to actually do our purification from. All right? So, because this is so important to our understanding of proteins, this purification, we always have to do this. A lot of research has been focused on how you do this. How do we purify proteins in large enough quantities to do experiments with, to study, or to use in industry. Nowadays, lots of proteins, enzymes are used in things like your laundry detergent or to make stone-washed jeans or something like that. So in order to use those, it's usually necessary for us to get it in relatively pure form. So there are two main approaches to this. The traditional approach is to purify the naturally occurring enzyme, the enzyme or protein that you want. If you know it's produced by a particular plant, you go out and collect massive amounts of that particular plant. We grind it up and try to purify the protein from there. That's one approach. Nowadays, it's more popular because it's a lot easier to do something called molecular cloning. Now, what this involves is isolating the gene that codes for that particular protein that you're interested in. There are fascinating ways to do this. So we can get a copy of that gene, and then we can put that gene in an organism that's a lot more easy for us to work with, like, say, a bacteria or something, and put it into that bacteria in such a way that we instruct that bacteria to make lots and lots and lots and lots of copies of this, huge amounts of this protein. And even better, maybe we can convince that organism to secrete it outside the cell. That makes purification much easier, one, because now the protein is not present in such low concentrations. It's the majority of the protein in the cell is this stuff. And the other is that if we can convince the organism to secrete it outside the cell, we don't have to do much purification as all. So nowadays, if it's possible, usually this is the preferred approach. But still with some enzymes, some proteins, it's necessary for us. If we don't know the sequence of the particular protein, we can't isolate the gene, we often have to rely on this initially. Questions on that? Okay. Before we even talk about, though, protein purifications, we need to talk about a couple of things before we embark on such an endeavor. One problem comes from the fact that these proteins can be inactivated. Usually that inactivation comes as a result of them unfolding. All right? So I've told you a number of times in this class that a protein's function depends on its three-dimensional shape. And during the purification process, depending upon the protein, it's actually fairly easy sometimes for us to unfold this protein, cause this protein to unfold. Many proteins, once that happens, it's difficult for us to get it to fold back into its proper shape. So it's inactivated. We call that denatured. Right? So many factors can cause a protein to unfold, such as drastic changes in pH. Inside the cell, pH is regulated. Once you break open a cell, now that enzyme or protein comes out in solution and it's susceptible to changes in pH. If the pH changes drastically, that can cause the protein to unfold and be inactivated. So whenever we carry out a protein purification, we're almost always going to carry it out using some kind of a buffer to maintain that pH at a constant level that will keep our enzyme in its active form. Another thing that can cause proteins to unfold is temperature. Higher temperatures can cause proteins to unravel, to unfold. So often, if we're carrying out a purification, particularly if we know that the protein that we're trying to isolate is susceptible to this kind of thing, we'll carry it out at very, very low temperatures. Carry it out on any steps that we do, we'll carry out on ice. Labs that do a whole lot of protein purification and have to deal with proteins that are sensitive to thermal denaturation like this might have rooms that are designed, they're just like laboratories, but they're a refrigerator. So I had the pleasure of working in some of those when I was in grad school as well. So it'd be 90 degrees outside and you show up to work in coat, hat, and gloves and everything because you're spending all day in the cold room. After I spent too much time in there, we also had an incubator room that was used to grow bacteria. So you'd oscillate between, spend an hour or so in there and then go stand in the incubator room. It's like 37 degrees centigrade. So do that too often and you tend to not feel very good after a while. Okay, so temperature is a problem. We can worry about. We can minimize that by carrying out these purifications at very low temperature. Another constant problem is that typically inside cells that we're trying to purify our particular protein from, there are other enzymes, and some of these enzymes are called proteases or proteolytic enzymes. These are enzymes that their job is to chew apart, to digest, to cut apart other proteins, usually fairly non-specifically as well. So obviously, we don't want these proteases chewing up our protein or hydrolyzing our protein that we're working so hard to purify. So what we typically do is, well, one, if we carry out the purification at low temperature, that's going to slow down the rate of the reaction catalyzed by these proteases. So that Temperature helps out with that. The other thing that we typically do is we usually, in our buffer solution, will add compounds that inhibit these proteases. These are compounds that bind to the protease active site and prevent it from binding to enzymes and chewing them up as well. Okay? So protease inhibitors. And last thing we have to worry about when we're purifying proteins is we want to avoid denaturation of these proteins by inactivation of them at interfaces. Proteins tend to unfold when they're at say, an air-water interface or something like that. So if you take a protein solution and you shake the heck out of it or something like that, or vortex it really rapidly, what you're doing is you're introducing foam into there, little air bubbles. Those air bubbles give more interfaces for that protein to become inactivated on. So we want to avoid any kind of foaming. Usually we're not going to shake a protein solution up or stir it really, really rapidly because we want to avoid inactivation at those interfaces. Okay? Questions on that? Okay, so next time we'll continue talking about protein detection and enzyme detection and then talk a little bit more about the specifics of protein purification.